Hi everyone! Today I would like to show you the results of our most recent research regarding the Andricat-Kaufman gene expression model. Let's get started. We'll begin with a brief explanation of gene expression. Then I will introduce the basic Andricat-Kaufman model, which was the starting point of our work. Then I will move on to our results and end with some conclusions and further research directions. First, let's get everyone up to speed on gene expression. It is a process which occurs in every living organism on Earth. It consists of two main phases, mainly transcription and translation. Transcription is the process of copying information from DNA onto a strand of mRNA. The DNA is split into separate strands of RNA, and then one of them is copied over to the mRNA. The mRNA is then used for translation. First, the bases are divided into groups of 3, each group corresponding to a different amino acid. The amino acids are then connected into long chains, which form the proteins. Those chains are then folded and undergo multimerization to form protein multimers. Why is gene expression important? Because it governs all life on Earth. And creating models can help us understand this incredibly important process much better. This is exactly why Andricat and Kaufman have decided to create their model. They started by taking into account the most important reactions taking place inside cells. Gene expression itself is represented by a single equation, where transcription and translation are joined together. First, RNA polymerase binds to a promoter, resulting in RNA polymerase, promoter, and a monomer of the protein. The monomers then undergo multimerization, where n units of the monomer connect into a multimer. Those multimers can then bind to promoters, blocking them and preventing further expression. This process is reversible, and single proteins can decompose into basic chemical substances. From these reactions, the following two-dimensional discrete time model can be derived. As we can see, it consists of two equations. The x represents the concentration of protein X in solution at a point in time t, and y is the same for protein Y. The parameters alpha, beta, and epsilon correspond to some physical properties of the genes and of the cell itself. Alpha is the rate of gene expression, beta corresponds to the decay of protein monomers, where higher beta means more stable monomers, and the epsilon is a coupling constant. Epsilon equal to 0 means the genes are fully independent, and epsilon equals to 1 means they are strongly interacting with one another. To simplify analysis, Andricat and Kaufman have made an assumption that the genes are identical, which means their rates of gene expression are exactly the same and can never change. To analyze more complex behavior, we have introduced a change by ditching that assumption. By allowing different values of alpha, we introduce one more parameter into the model, and at the same time extend the upper bound of alpha, since there are no known physical constraints limiting alpha to the range of 0 to 100. Before starting any numerical simulations, we wanted to check that the trajectories of the system are bounded. To do that, we calculated the absorbing set of the system and have shown that under all application-relevant circumstances, the system is in fact bounded and simulations can be accurate. A set is absorbing when, for any given starting point, after some finite amount of iterations, the trajectory ends up within that set. We have shown that absorbing set exists for the Andricat-Kaufman model with our modified assumptions, and is in fact a rectangle, the dimensions of which depend on the model parameters and some arbitrarily small constants C1 and C2. Knowing that the system is bounded, we can move on to numerical analysis. Here we can see bifurcation diagrams made for the original unmodified Andricat-Kaufman model. Despite the simplifying assumptions, we can see that the behavior is highly complex, and there are regions of stability and chaos, with transition to chaos occurring through a series of period doubling bifurcations. Now let's compare that to the model without the simplifying assumptions. To create those diagrams, we have fixed alpha2 at a value of 200 and varied alpha1 to record the x and y coordinates of the attractors. We can see a significant jump or discontinuity occurring at alpha1 equal to 200, which is the value of alpha2, which, as we will show further in the presentation, results from Bistability being created in the system. This next diagram is created by varying both alpha1 and alpha2 along a linear segment of the parameter space. Here we can see a wide landscape of different behaviors of the model, which are remarkably similar to the behavior of a logistic map. Knowing that both versions of the model could be chaotic, we wanted to check whether one of them was more chaotic than the other. To do that, we scanned the parameter space in search for parameter combinations resulting in chaos. Blue indicates regions where chaos has been observed in both models, while orange means that chaos has only been observed in the modified model. Looking at those figures, it is clear that removing the simplifying assumption does not significantly destabilize the model. To identify chaos, we have been relying on Lyapunov exponents. Lyapunov exponents are numbers which help quantify chaos. Positive Lyapunov exponents mean chaotic behavior present in the system, while negative indicate the existence of a stable orbit or a fixed point attractor. This figure presents the values of the maximal Lyapunov exponent for different combinations of alpha1 and alpha2. We can see alternating bands of chaotic and stable behavior. It is worth noting that this graph is symmetrical because it inherits the symmetry from the equations defining the model. This, however, is not always the case. If we focus on a single initial condition fixed somewhere in the phase space, we can see that symmetry breaking is common. It turns out to be the result of Bistability, which means that two or more attractors can coexist next to one another. Upon closer examination, we have found a variety of different types of Bistability. Here, we can see a stable cycle right next to a chaotic attractor. Other types include two or more stable cycles or two chaotic attractors. Such kinds of Bistability have never been described before for the Andricat-Kaufman model. To identify parameter values which create this behavior, we utilized the symmetry breaking mentioned earlier. We introduced two metrics. The P plus minus is the proportion of alpha1 and alpha2 combinations, which result in both positive and negative Lyapunov exponents depending on the starting conditions. S prime, on the other hand, is the Euclidean distance of the Lyapunov exponent matrix visualized on the previous slides from its transpose. Both of those metrics assume higher values when symmetry breaking is present, which indicates Bistability. Those findings show that the Andricat-Kaufman model can potentially simulate real phenomena such as genetic switches or cells stabilizing and destabilizing based on even gentle external stimuli. More extensive computer simulations could be used to create a multidimensional map of all possible behaviors, which would help estimate the probabilities of encountering Bistability, chaos, and order. A much more in-depth explanation can be found in our paper. Thank you for watching.